Thank you, everyone, for returning back for the test session, rather. And I hope you all had a good break. So we're just gonna go straight to the session 2. Basically, we're gonna do something a bit different. So, Okechukwu, I think you'll be speaking this as you'll be the second speaker because Ashraf had a lecture, and I think he was thinking previously he was going to speak tomorrow. So the next item on the agenda would be Professor Ashraf El Alali. I'm just going to pull up the Professor Ashraf's so Professor Ashraf El Alali, he's the computational biology and associate professor at Mohammed VI Polytechnic University in Morocco. He's going to talk to us about Africa-based databases to improve agriculture, One Health, and biodiversity. And I basically would let you all know that this is going to be an interesting talk. Ashraf has been doing a lot, especially on the African continent, when it comes to databases, annotations, and it will be an interesting talk. Over to you, Dr. Ashraf. I will make you a co-host just now. You can share your screen. Yeah, you are now a co-host. Please unmute.
Okay, now I'm unmuted. Thank you, thank God, thank you. And apologies, I had this on my agenda for tomorrow, but unfortunately, just realized it's today. So let me just share my slides as I'm saying this. Okay, can you see my screen?
Yes, please, carry on.
Okay, all right. So thank you for having me. I'm Ashraf El Alali, professor at UM6B, computational biologist, bioinformatician. So today, I'm going to touch a little bit about the different databases and tools that we've developed and how they could reach to this One Health and Biodiversity and how it all relates to the mission of AfricaBP. So just a little note about where I'm from. So Mohammed VI Polytechnic University. If you haven't heard of it, it's an international higher education institution established to put research up front and innovation. So we have a vision of driving eco-social economic development through applied research and innovation, not only for Morocco, but for Africa. So this is a big mission. So we are obviously doing our part of, among many other institutions in Africa. So as part of the university, I am the head of the Bioinformatics Laboratory, which is part of the College of Computing. So the Bioinformatics Laboratory conducts research in the fields of bioinformatics, computational biology, computational chemistry, and it's meant to serve not only agriculture, environment, but also the health sector. So UM6P, we have state-of-the-art NGS sequencers, we have mass spectrometers, we have also host Africa's fastest high-performance computer and largest high-performance computer. And so our goal at the bioinformatics lab is to leverage the data coming from these NGS sequencers and the available compute to drive the needs of UM6P, the need of Africa to develop innovative research and training programs in bioinformatics and computational biology. So we have four areas of research. We do applied machine learning to different omics, whether it's genomics, metagenomics, different kind of omics, and integration of these omics approaches. We look at genetic markers in plants. So this is kind of where we would be talking today. I don't know if you see a pointer. Let's see if I can use a pointer. It's going to take some time. Here's the pointer. Okay. All right. So the pointer works. So we do GWAS studies, whether it's at the agriculture or even at the medical level, and we do molecular simulations in the drug discovery. So these are four different main research areas. Today, we will concentrate on genetic markers and particularly on databases and tools within these approaches. Okay, so we have a plant bioinformatics database accessible via our website. We'll share the link towards the end of the slide or within each tool, and it will be available at the end of the last slide. And so this bioinformatics platform is meant to be both a data hub, hosting data sets that are either computed from local data or that are generated as a result of analyzing existing data sets. We also have several genomic tools, and the idea is to have to reduce the bioinformatics burden from some biologists, agronomes, people who use genomics as a tool rather than being specialists. So we try to alleviate that for several different pipelines and processes. So our platform is accessed, like I said, worldwide. So here is just a quick summary of our statistic page. So we have over 52,000 visitors so far. We've been operating for about almost 2 years, a little bit less than 2 years. We have visitors from these are the top 10, for example. They go from the US, India, Germany. Within Africa, we have Egypt as our biggest visitor. And we look forward to actually serving. We do serve all countries, but this is we have some users from South Africa, from different countries around Africa here. But these are just the numbers for the top users. And then, of course, we have some statistics on how many people actually used our tools, run actual jobs for the last 3 months here, and how many people have downloaded some data from our databases. Okay, so I will go through some of the examples of these tools that we have, and then you can see the full platform on our website. So the first one I want to go over is plant LTRDB. So transposal elements are genetic elements that can move or transpose from one location to another within a genome of an organism. They are known as transposons, or what if you've heard the word jumping genes. So there are two main types: DNA transposons that do like a cut and paste, while retrotransposomes moves by copy paste. So that's the difference between them, but they are all referred to as transposon elements. They have different, they create genetic variations and drive evolution. especially when they introduce new genes or they alter the gene expression pattern. And we have published some papers showing the importance of transposer elements specifically for plants. So the issue is, they are not easy to, the pipeline to do so is not as easy to find, um, to actually run on your own, and it requires a lot of steps and further post-processing by in-house code. So we have this database that has all the access 195 plant species. This number comes from the available as of 2023, the available plants that have full annotations, within the NCBI. Because we don't just find these LTRs, we classify them, different classes, different clades and things like that, and also how they relate to genes, because that actually changes. They can disrupt gene function, they can cause different variability. So we want to actually give the user information about how they are within relations to the genes of that particular plant. That's why we only processed here the plants that do have annotation. And then, of course, we classify them. This is kind of the different approaches for LTRs, the different elements that exist. And based on how many elements are present, these LTRs have different classifications. You can get that from our database. For all the elements in the database, you can view them on a map. You can download the data in different formats and things like that. Now, not all of since 2023, there has been a lot of different plants that have been posted. So instead of actually updating the database each time, we have actually opted of making all our code available as a tool for anybody who has sequenced a new plant genome. So since our publication of plant LTR-DB, so now we have a tool called Mega LTR. This tool actually computes, identifies, classifies, LTR, intact LTR-RTs for whole genome level, and integrated different tools that are structure-based, homology-based, de novo identification. All of these looks at the different tools and comes up with a consensus among all these different approaches, and then it does classification based on which type of LTR, and then annotation based on how close is it to a gene or not, gene chimeras, and so forth. So a lot of different analysis that one can run. So also we provide statistical analysis, visualizations, insertion time, as you can see here, visualization of these LTRs within the chromosome. So these are actually visualization that we have created ourselves here. And for each genome, you get figures. We see our figures showing up in a lot of publications now. So some people take, once they annotate their genome and they run our tool, they take these figures directly into their publications, and we're happy that we started seeing this now. Plant LAI. So LAI is an index that can identify it's an LTR assembly index. It has been used recently as a quality check for genome assembly, particularly for plants. Okay, so the higher the LAI value associated with higher quality of assembly, and there is like so there is a specific range that you can tell that this is a reference genome, or this is a low quality, or this is a gold standard, and so forth. All of that can be found in our publication. So we assessed about 1,644 plants and altered genomes, and we've pre-computed it, because once this LTR LAI came, anything that was submitted before, people don't know how good this plant genome is. They can still access the traditional, scores of quality, but for plants, LTR or LAI value gives a much better understanding on the quality of genome assembly. And so we've computed all of the available genomes at the time that were of good quality. So there are more at that time, but they were so low quality that LAI is not even valid approach. So we've taken all the ones and we've computed it, put it in a database. People can access it, reach the data. ask for, given particular, you know, genome ID, and then they can get the scores. We have some visualizations as well. But again, the same thing we did once we have actually created this tool. Now others will say, well, once we have a new genome, it's not in the database. So as we did with Mega LTR, instead of having to update this database every time, we are actually allowing there is a tool here where somebody could put in their newly annotated, assembled. It doesn't have to be annotated. Once you assemble your plant genome, you can come to this website. You can click on, upload it to our database, and you will get upload it to our web server, and you will get the LAI score. Now, these are actually all run on high performance computing. These jobs take days to run. So these are not your traditional web services that you will see run in small jobs. So things, go into the Africa's largest supercomputer. They get computed, and you get that in a few hours instead of a few days. For large genomes, it can take up to 7 days. If you put wheat, for example, it will give it to you, but it will give it to you about 5, 6 days later using 56 CPUs, which is our default value for what we're given here within our service. Okay, so we're not only hosting the data for existing genomes that are already published, we're also allowing new sequencers, new assembled genomes to be able to compute this score as easy as uploading it to our server. We also have comparative genomics. So we do, for chickpea, we have a website that compares these different, not just LTR, but all types of transposer elements within different types of Sicer species. So that is also available on our data. It's has the same visualization tools, classifications, all of the things I've mentioned before, but now for all transposal elements, not just LTR RTs. We also looked at, how can these genetic markers be used as in actual plant breeding programs, in actual laboratory work. So we have looked and surveyed the database, the literature, and found, all the people that have used certain transposer elements as markers, and then we are made them available in a database. You can search by primer, you can search by type of transposer elements, and you get all the all what you need to do your experiments. Now we also have another tool called Mega SSR. So SSRs are simple sequence repeats. They are also used as genetic markers. This is one of our #1 used tools. This tool gets run, and I want to say 100 times a week minimum. So it's SSRs are one of the easier genetic markers that can be applied in the labs because of the size of the SSRs are not much smaller than LTRs. So you're covering a smaller region, and so they're still the favorite genetic markers. So that's why it's this particular tool is more used than others. But we expect as technology, as NGS gets used more, people will start looking at LTRs as well as genetic markers more as well. So we have a tool that identifies all the SSRs, classifies them, allow you to download all the different statistics. You can view them on our website and query the results on our website, or you can download the full output and do whatever analysis you want with it. We have a primer design. So actually this has an in silico primer. So once you've find your SSRs, you can identify which ones you want to actually do in silico primer. And then you can actually check that these that you will, what kind of bands you would expect to get if you were to run these particular primers in in the lab. So it designs the primer for the SSRs that we identify, and also we have a tool to allow it to kind of do in silico PCR validation. Okay, to just get an idea of what you expect. Of course, it's not going to replace your actual PCR. Contribution to research. Of course, now this is going to accelerate plant species research. This was very, in our workshop, North African workshop, people really, appreciated this tool that were, especially those people who were actually doing plant breeding, needing genetic markers. They were very happy to learn how to use this tool. And again, based on our feedback, also on the number of jobs that are run, this remains one of our top tools. We have also a database of for all plants of tRNAs. So tRNAs are non-coding RNAs that are essential for protein translation. So they're important to know. They differ from one species to the other. So we have identified them for all possible plants databases that we could found as of 2022, and they are available as well as all kind of visualizations. You can actually blast against them if you have your own tRNA. You want to see if it exists in which plant. You can blast against our database, or you can download the tRNAs per species, per type of tRNA. So nobody can have to compute this on their own. It's all available in one of our databases. We also have pathways pre-computed for, we have over, 2.7 million different pathway based for all the available plants as of the publication, which was back in 2021. So it's all available as well here. For each plant, you get all the different pathways. You can actually interact on our maps and select which pathways you want to concentrate on, add new genes and things like that. I'm going to get a little faster because I'm running out of time. We have also a line stats plot. This is a tool that does a lot of visualization and actually computes different multiple sequence alignments, post multiple sequence alignments processes. So once you've aligned your sequences, generating, doing different PCA, post MSA analysis, different things that one can do by just giving the your MSA file, or even just the raw data. We do all the steps. We give you all the different clusterings, the trees and the heat maps, PCA, everything gets downloaded by one. So that kind of alleviates this because this is a repeated analysis for a lot of different researchers. So we thought we make our tool available as well. So it gets used pretty often. All right. So all of this brings us to what I wanted to really talk about is our vision at AfricaBP is of creating an African DSI data bank, kind of similar to what is available at NCBI or EMBL or the Japanese version as well. So we want to have our own, not similar, not a copy, our own envision, our own way of doing it as well. So we are looking at having a federated platform, which would be a network connecting African institutions for different DSI storages, analysis, visualization, and sharing. And then, of course, the institution will retain their own data sovereignty while adhering to their own regulations. But we somehow have a federated platform that can allow access to these different data sets that might reside in different databases scattered across different institutions. We're also looking at having this what's called the global access point. So this would be a decentralized network providing access to sequence data, metadata, and also access to analysis as well. Different analysis, HPCs around Africa that may offer some sort of computational help to different researchers in Africa. So of course, we all know the challenges. We have fragmentation data. Our data is just remaining in different databases. There's a gap of not having enough HPC and computational power for many researchers. And so the impact remains of these data remains, remains unseen unless we actually have this, DSI that will actually alleviate some of these issues. Sorry if I'm going fast, but I think I'm gonna run out of time if I don't. So a key component of this African DSI, we want to have a platform, a database platform to integrate all the existing, different DSAs, as I've mentioned before, and use the federated model to overcome any regulatory challenges. We also want to have a data analysis platform. So this would be a cloud-based or access to HPCs, whatever available, or through bioinformatic tools and web services like the one I host here in my lab. Visualization platform, so allowing people to actually visualize their tools, such as the ones I've mentioned that we host as well, making complex genomic data accessible for analysis and interpretation. And of course, sharing the data, being able to share this among different, among researchers, but being compliant with the national and global regulations. So we want to create this federal database platform by linking existing DSIs, integrate data, by analysis platform, establish data visualization tools, promote responsible data sharing, and engage policymakers. This would be the strategic priorities for us in this effort at AfricaBP. Roadmap. First, we have to build the network, expand the capabilities, and have some long-term goals. I think I will just leave some time for questions. We know that nothing happens without having funding, so we have to, we've laid out some funding mechanisms and government's frameworks. All of this is available on a preprint that you can see, that we have shared at AfricaBP a few months ago. We want to have some, all of this has expected outcomes, enhanced biodiversity conservation, which is one of the main goals of AfricaBP and all our efforts as researchers and genomic researchers in Africa. Strengthen research capacity, so we want to actually increase collaborations among African institutions, improve access to the different tools, different computation among African institutions and collaborations, and of course our support for food security and sustainable agriculture by using genomics in the way it's meant to be used, to improve agricultural resilience and biodiversity conservation efforts. Of course, we call among you to help us engage. Researchers are one of the main stakeholders. If there are any policy holders among us here, institutions, private partners, we call on you to join us with this effort. You can reach, of course, AfricaBP to know how you can help. Okay, thank you very much. This is the website for my lab. You can access the platform through this, and then these are some emails if anybody wants to reach out. And thank you. Sorry, I had to speed up at the end.
Thank you, Ashraf. That's an interesting presentation, and also very interesting to see the number of users. So, I mean, I have a question, but I'm just trying to see if there are speakers who participant on Zoom or on site who have any question. Do you have any participants on site with a question, please? So I can see Hewan Degu. Please ask your question on mute.
Okay, thank you very much for giving me this chance. I'm really happy and pleased attending Professor Ashraf's lecture. It is really a wonderful job, specifically working on plant. As you know, we have a lot of indigenous plants and which helps us to secure food. And even if, like, we do have a different diversity, root and tuber crop from the western part of Africa, and also cereal in the northern part. And also, as you know, in Ethiopia, also we have a cereal and also multiple crops, including trees and others. And I'm really happy I am learning the great job that you are using. I hope that we are going to access the tools that you have really developed. It will help us because we are going toward this indigenous crop sequencing and transcriptome analysis. Because as you know, in Ethiopia, one of important resilient crop is insect. And that is where we are using here in the southern part, where the Sidama region and specifically here in Awasa. So that will be a great asset for future collaboration. I think I will communicate with you if you are really willing, Prof.
Welcome.
Okay,
Most
and
welcome.
that is, yeah, so definitely I will disseminate all this information to our university communities, and we'll look for a future collaboration. I'm more than happy to be here. Thank you very much, Prof.
Thank you.
Thank you.
The platform is available for all the different annotations. And if you need any other, annotations, any other pipelines, we're welcome to collaborate on those as well.
Thank you. Thank you, Awan, and also Dr. Ashraf. So, is there any other person that have any question on Zoom before we go to participants on site, please? So, while we're trying to see if there are more questions on Zoom, is Professor Elali, some participants are asking if your presentation is going to be available for them.
Sure.
So,
I can definitely share it.
Yeah, just share it in the chat box if that's okay.
Sure.
Just feel free.
No problem.
Yeah. Then all right. Okay, participants on the Zoom, please.
Thank you very much.
Ashraf, if you can stop sharing, please.
Stop sharing.
Yeah. Yeah, please go ahead.
Okay, my name is Dr. Emmanuel from Federal University of University States. I'm really interested in the plants because I'm doing some work using a kind of organic water to work on import community. But the question I want to ask is that is there any way we can do the genomics of some of those plants to know the bioactive substance that can bring about curative effects in some ailments like cancer? So this is actually what I was. Thank you very much.
Thank God. Did you hear?
Yeah, so Professor Julian, we didn't quite catch the question. Could you please maybe ask it in a different way?
Let me say what I understood.
Okay, all right.
You're asking if there is any mechanism to help plants become more resilient for something specific? Is that what you're...
What I'm saying is that is where we can actually, when you are doing the plant study, you do some phytochemical and phytohormones to know some substance that are there by active substance that will cause some curative effects medically. And so what I'm asking is that some of these active substances, in any way, since you are doing some plant genomics, gene markers, trying to identify some genes in the plant, is there any way we can identify some genes in the plant that are responsible for the curative effects that those plants contain?
Yes, okay. So that would be the GWAS approach. So we look at different phenotypic information as in response to different traits, genomic traits of or genomic markers of that we have. So that is possible. We can also look at comparative genomics, comparing the different even within, for example, LTRs, which is what takes a lot of parts of my work, looking at different, the way, chimera genes have certain LTRs that are causing this traits to be available in this plant versus this plant. So there are different known approaches. I just mentioned a couple of them. I don't know if that answers your question. I'm sorry because I didn't hear it.
Yeah, so I think to also add to that, things like the typical annotation work could do, like gene expression as well, right? Where you could find, okay, what genes are responsible for X, Y, Z, and comparative genomics. So there are multiple ways, essentially. The GWAS approach, as you mentioned, there is also the annotation, or even the comparative genomics approach, where
transcriptomics would be
transcriptomics as well, right? Gene expression, looking into that.
Or even looking for markers of, if this trait happens in a particular species and this could be also possible that it's available in your species, in your plant, you can look at these, use the markers, the SSR markers, LTR markers, to see if they exist in also in your particular plant. So that kind of